engineering technique of DNA isolation and determining its sequence. It was ambitious mega project to sequence the whole genome of human. The goal
Right.
of this project was number one, to identify all approximately 20 to 25,000 genes in the human DNA. Secondly, determine the
Yeah.
sequence of 3 billion chemical base pairs that makes up human DNA.
Yeah,
To
but
store
at
this,
that point of time, we did not know these numbers, right? Remember that?
yes,
So
yes.
they're now written as goals because now we know what came out of it. But I think what is the best way to learn is that goals are set before you begin anything, right?
Yeah.
So instead, if you write, identify the total number of genes in human genome, which later was found out to be 25 to 28,000. Determine the total sequence of the human DNA, which later was found out to be 3.3 billion, right? Yes, continue.
Yes.
Just making sure that you know these things.
Mhm.
Yeah.
Then to store this information in database, to develop, to improve tools for data analysis, transfer related technologies to other sectors such as industries. Fourth thing, address the ethical, legal, and social issues that sectors such as address the ethical, legal, social issues that may arise from this project.
Right.
Then the Human Genome Project was a 13-year project coordinated by US Department of Energy and the National Institute of Health.
Yeah, not
There were
just
numerous,
coordinated, they also funded for it to begin with.
yes, there were numerous countries participating in the project. The Wellcome Trust UK became a major partner. Additional contributions came from Japan, France, Germany, China, etc. The project was completed in 2003, and the sequence of chromosome 1 was completed only in May 2006. This was the last of the 24 chromosomes, 22 autosomes X and Y to be sequenced.
Yeah.
Then let me discuss the salient feature of human genome. The first
Yes.
human genome contains 3,164.7 million base pairs. The
Yeah. You
average
can
gene
just remember it as 3.1 billion. It's a better number too.
yeah.
Yeah.
The average gene consists of 3,000 bases, but size varies greatly, with large known human gene being a dystrophin at 2.4 million bases.
And
Um,
that's the largest known.
yes, yes.
Yeah.
Then the functions are unknown for over 50% of the discovered gene. Less than 2% of the genome codes for protein.
Yeah.
Repeated sequence make up a very large portion of the human genome. And chromosome 1 has the most gene, that is 2,968, and Y has the fewest, that is 231.
Perfect. Right.
Yes.
So you could understood it very well and in detail as well. So, and what do you think? I also asked you to go and to read the application and future challenges, right? So you started with a right note that first thing is whenever you start something which is being started for the first time, it opens new avenues for many research sectors and disciplines to grow within it, right? So Human Genome Project was not just one standalone project that you started, you found something and you shut it down. It impacted the course of fundamental research, evolutionary research, genetic research, even medicine, drug designing, everything, right? So
Yes.
applications are enormous. Once you know human genome, it enables you to start a new approach in biological research. And earlier, like when people did not know about the whole genome, they used to study a single or a few genes at one given time, right? Like suppose you found a mutation in one part of the chromosome and you will just study that part of the chromosome. But once you know this human genome, you can study a widespread effect of any phenomena, chemical mutation, or a process or developmental defect on overall genome. right? So with whole genome sequences coming out, remember I told you when this project was funded to begin in 1990, it was estimated that somewhere we will end up using $9 billion. And they gave a green signal to it. Okay, let's do it. But 13 years is a big time. And including chromosome 1, it became 16 years. It was a big time. In 16 years, a lot of technology started growing and became high throughput. So cost went down. And by the time this project finished, it finished well within just $3 billion. Somewhere around 2.7 to 2.8 billion dollars were used in the whole project, and it was finished. So that also tells us that when we have very long, ambitious projects running, what we estimate at the beginning and what we end up, everything becomes better, easier to make, and high throughput with time. Correct? Look at anything around us, like the cost comes down for manufacturing or doing that process because technologies evolve. So same happened with Human Genome Project as well. And another important thing is that now on a very broad scale, people could study multiple genes in a genome. For example, you could even study all the genes in a particular tissue. For example, if it's a specific kind of a liver disorder, liver cirrhosis, liver cancer, or lung issues, or any other organ issues, you can study all the genes in that particular tissue or organ, or in a tumor, okay? And then see how they are interconnected to each other, how one gene misregulated or mutated affect the expression or activity of other genes as well. And this could only be possible because, yes,
So what's tumor?
a tumor is just uncontrolled growth of cells. So you know that our body is divided into, on the basis of origin, it's divided into 3 germinal layers. So everything in the body either came from the mesoderm, endoderm, or the ectoderm, right? Certain
Yes.
organs came from ectoderm, like our skin and the brain and the nervous tissue. And many organs which are inside our body came from the mesodermal origin, like our heart, etc., and also endodermal origin. So when cells of a particular tissue or origin starts dividing, and they lose this property of inhibition, contact inhibition. So when cells come in close contact, they stop growing further because they know that there's no further space to grow. And whatever they wanted to achieve, they have achieved it. For example, there is a injury in the skin. So we got a cut in the skin, and then skin starts to regenerate. But when the wound is healed, the regeneration stops, right?
Yes.
Because it has to fill for the missing cells. Similarly, inside the body also, for repair, wear and tear, many a times stem cells divide and proliferate and then stop. But in case of tumor, they do not stop. They forget to stop and they make a lump there. Okay.
That's due to
And if
mutation?
it can be because of mutation. And if this tumor has the property of metastasis, like it can dislocate from one particular place where it was growing, and it can go and cause a tumor in other part of the body, then it's called a cancer. So cancer is a metastasizing tumor. It's called a malignant tumor. A tumor which just grows in one place and stays in one place and does not, it grows to a point and stops and does not spread, is called a benign tumor. That's non-cancerous benign tumor. Otherwise, if a tumor has the ability to spread, it is called a cancerous malignant tumor. Malignancy is the property of tumor to translocate to different parts of the body. Is that clear?
Yes.
Yeah. So now people are doing these whole genome studies on these tumors, that what is exactly that has changed in that tumor, like how many genes are going up and how many genes are going down, and all that is possible because we know the whole genome sequence. Correct? Now, I also asked you to go back and look at the methods. Did you look at the methods? Methodologies of how they were sequenced? Did you look at that? Yes. So would you like to just tell me about it? And if anything you miss, I'll just pitch in again. I'll add things. So let's start with methodology.
Yes, sir.
Okay,
So this.
Mariam has joined us. Good evening, Mariam. Okay, so Iram is kind of enlightening us about methodologies that were used to sequence the human genome. Yes, Iram, over to you.
Yes, sir. So this methodologies involves two major approaches. The first one is ESTs, that is Express Sequence Tag, and the other one is sequence annotation.
Yeah.
Yeah, so in Express Sequence Tags, we identify the genes which only express in the form of RNA. And in
Right.
sequence annotation, we extract the DNA and study the sequence by the method called as Sanger sequencing.
Yes.
Yeah.
Yeah, correct. So one thing that is very, very clear from these two methodologies is that expressed sequence tags, can I call it targeted sequencing? Because it's more focused on all those parts of the DNA which can produce RNA, which means the part of DNA that can get transcribed can only be sequenced through this method. It's called expressed sequence tag because you use a tag, okay? And that tag is that when that part of the DNA gets transcribed, you get that tag also in the RNA that is produced. Okay. So the first approach, which is expressed sequence tags, because the sequence is expressed as a RNA, is called targeted sequencing. Is it clear?
Yes.
This will only tell you about the genes because genes get transcribed into RNAs. I'm not talking about only the protein coding genes, even the non-protein coding genes, which can get expressed as RNAs, like the rRNA and the tRNA and others, which do not form a protein, they will also be sequenced through this technique. So the only criteria is that it should be expressed as RNA, right? The other one, so this is more targeted. Just write it down. Sequence targeted. Okay. The other one is called a blind approach. Why it's called a blind approach? Because here, and why this is also important, because I told you not all the DNA in the human genome like codes for protein or even for RNA, right? There is a whole huge bulk of our DNA that does not get transcribed or translated, but is still sitting there. Okay, earlier we used to think that it's junk DNA, but it is turning out to be not exactly junk. It has some regulatory roles. Okay, not all, but some major segments have regulatory roles. Anyways, but in this case, it's called a blind approach because you are simply just sequencing the whole set of genome, all the coding parts, which are coding for a RNA and a protein, and all the non-coding sequences as well. Now, how do you do that? First few steps are simple and are common to both the methodologies. The first step is that you have to isolate the total DNA, extract the total DNA from a cell, right? So you take human samples, their cheek cells, their hair follicle cells, or their blood cells. You can take any cell that has any sample that has live cells and contains DNA, and then you extract DNA from these cells. And DNA extraction you already have done also, right? As a part of your experiments in biology from, I think, banana or some fruit, correct? You have isolated DNA in your schools, Iram, Maria? Iram, I could not hear you.
No, sir.
No, you have not done DNA isolation yet, and as a part of your experiments from a
No,
banana
not yet.
pulp or something. Okay, so probably you'll be doing it soon. So I think in biotechnology they teach you how to take out DNA. Probably you have not reached that point, but DNA isolation, that's a crude way to do DNA isolation that you learn in school, but the principle is same. You break open the cell by using some detergents. Yes, Iram.
So how do you extract the DNA from the cell of a blood, that is RBC, which do not have any
Yeah,
genetic
we
material?
cannot extract it from RBCs, but blood also have WBCs, right? So we extract it
Yeah.
from WBCs. Yeah, WBCs have nucleus. So yeah, and yeah, so WBCs, not RBCs. So when you're taking blood sample to extract DNA, that will come from the cells that have nucleus, which are your white blood cells, not the red blood cells. Or you take a cheek sample or hair follicle or other samples as well. Anyways, so what I was saying is, yeah, the concept remains the same. You have to break open the cell, then you have to break open the nucleus, and you have to precipitate DNA. Now, about DNA, you know a lot of things by now. So tell me some property of DNA that we can use to precipitate it separately from every other component in the cell. So when you will make a juice of the cell, it will have protein, lipids, everything, lots of things in it. How will you just specifically extract out DNA? Any property you remember about DNA? We studied a lot of properties of DNA. Anything that can help you selectively? Centrifugation, but how will centrifugation helps? You're right, centrifugation can also help you, but it's later on. So once you know that your sample has DNA and most of the things are lighter than DNA, then you can centrifuge it and precipitate it down. That's there. But how will you target it to come out from the cell solution without a protein, lipid, and other impurities along with it? Because you don't need proteins and all, right? If they come in your sample and you try to sequence it, it will be a problem. You just need pure DNA to sequence. So DNA isolation is a very, very tricky and very, very careful step in this whole process. So remember I told you that DNA is one, it is negatively charged, right? So if it is charged, it is then it is negatively charged. So it can be precipitated and taken out from the aqueous solution, which is the cytoplasm, if you provide a layer of super chilled ethanol. Okay, so ethanol, which is a carbon-based organic compound, if it is chilled, DNA just precipitates in that layer, and that's how we extract DNA out of. You will do this yourself in your biology labs. You will extract DNA from banana fruit, because that's easier. But once you get the DNA, so this is the first step. Write down the total DNA from a cell or a tissue is isolated. But when you will isolate DNA, you will realize that all these DNA clump together and forms very, very long threads. So you basically spool DNA out from your solution. When you will do it in schools, you will see that. You will never see a single DNA thread. It's not possible. You will see a clump of DNA together as a gluey thing in your solution. But we cannot sequence that because those are first multiple DNA threads and second very long polymers. So we have to fragment it randomly. Okay, so we use restriction endonucleases, right? These are the enzymes which cut DNA, restriction endonucleases, and we make random fragments of small sizes. Okay, and this is easy to sequence now. But the problem is, once you sequence a fragment, how do you know where this fragment fits? So for that, these fragments have tags at the beginning and at the end. It's like, have you solved a puzzle, like these picture puzzles that come?
Yeah.
So how do you know which piece goes where? One is you try to see which piece goes where, but sometimes for help, they have an extra bulge out or an indent in some pieces, right? So one bulge fits in the other indent. That is also one way to know that, okay, this piece will go here. You understand what I'm saying? So let me tell
Yes.
you. So one piece, let's say, is like this. So you will have one piece which will be like this, right? So, you know, oh, this piece can fit here because this has the same indent. And then the adjacent piece can have an indent and can have a piece like this. So you know that to fit this piece, we need someone which has an indent like this, right? So this piece now goes here and a piece similar to this comes here. You understand what's happening? And this is how these are helpful tags, right? That help you to fit pieces together. Do you understand, Iram and Mariam?
Yes.
So similarly, when we make fragments using restriction endonucleases, there are some tags at the beginning and the end, which helps us, and this cannot be done manually. So computer softwares helps us to align them. That's called alignment of the sequenced genome. So they are overlapping fragments. So we always generate overlapping fragments. These overlapping fragments are easier to align on the basis of repeating sequences or overlapping sequences. And that's how, after sequencing small fragments, we can stitch them all together and say that this is the human genome. Otherwise, that's not possible to do. Yes, Iram, you had some questions, I guess. You unmuted. Do you have any questions?
So when you dissect the whole long polymer of the DNA, then every fragment will be similar, right? So why do you need sequence tags for it?
Each fragment will be similar.
Yeah, like cutting the DNA, it will be similar, right? I hope you understand.
So what? So you mean? I think what you're trying to say is that restriction enzymes have a particular site where they cut, right? Are you talking about that?
Yes, yes.
So yes, you're right. So but sequences will not be similar. So let's take a sequence: CACATTAGCCTAACCG. Okay. Now let's say this is a sequence. You are randomly cutting it, and you have used a restriction enzyme that cuts between A and T. Okay. So one place it can cut is here. One place it can cut is here, right? Let's say. And if there was one more, let me make it T for some reasons. So one place it will cut is here, right? Now, this sequence, and this sequence will not be same, right? Restriction endonucleases will cut at random places where they will get the recognition site. Now the recognition site is mostly 4 to 6 base pair, minimum 6 or 8 base pair or 4 base pair long. I'm just giving you an example. But even if it cut at multiple sites, do you think that this sequence is same as this sequence? Even the length is not same, even the sequence is not same.
Yeah,
right?
yeah, I get it. Yeah.
So how will we say that this was 1 and this was 2 and this was 3? Once you have cut it, what mistake you can do is you can take this as first, this as third, and this as second. Then you will mess up the whole genome. So that's why you need overlapping. So how they will cut is like this. So let me give you an example. So let's take the same sequence and let's see how cutting happens. So in the same sequence, what restriction enzyme will cut at so one fragment will have CACATA. This is one fragment. Then other fragment will have TTA this, then other fragment will have be this, and then other fragment will be this, right? So this is fragment 1, 2, 3, and 4. Now you can see in all the fragments, there are at least 3 overlapping things, right? Do you see here overlapping?
Yes, sir.
Now, based on this overlapping, you can tell that, okay, if I give you sequence 2 first, so sequence 2 is TTA, GCC, TAAT. So you got this sequence done, this fragment sequenced. Now you don't know which is this. Then you got this then you get this first one, which is CACA, TTA. It is smaller than this, but you got it. And you still don't know where to put this. Then you got the third one sequence, which is AAT, TAA, GGTCA, right? Now, look here. So when you got this fragment first, you will put it in computer along with these 2 fragments, okay? Now, what computer will do is, it will look at the ends. It does not care about what's in the center. It will look at the ends. So when it will look at this end, it will see TTA. And here it will look at AAT, right? Then it will look at sequence 2. It will find CAC and TTA, okay? So this has TTA and this has TTA. You understand? Then what it will do is, it will try to align it like CACA, TTA, and then TTA, GCC, TAAT. It will keep this as 1 and this as 2 and merge these because this looks same, right? This is flanking sequence. So the sequence becomes CACA, TTA, GCC, TAAT. Is it the original sequence? Yes. Then it will look at the third one. It has AAT and it has TCA. Does anything rings a bell? Yes. So you see AAT here and you see AAT here. So this ends with AAT. So computer will just align here AAT. This looks like same. And then whatever is further. So similarly, it will keep aligning and keep joining ends on the basis of overlapping sequence. Do you understand? That is why it is important to have overlapping sequences. Does it make sense to both of you?
Yes.
Yeah. If it does not make sense, we can do it together. No problem. But you should understand it. Should I go over it once again quickly? Or it's clear? Tell me, people. Just look at write this in your notebook and look at these 4 fragments which were generated randomly. And then each fragment looks something like this individually. And on the basis of the first and the last few, you can align it. So you see that this fragment starts with TTA, where this fragment ends, TTA. So maybe this TTA is common. So this fragment, this part comes before this here. That's why TTA is common. And this fragment ends with AAT. The third one starts with AAT. So maybe this whole thing comes here after AAT. Do you understand?
Yes, sir. It's clear.
Yeah. So this is how the whole genome is stitched. And it is not just done once. It is done multiple times, like even hundreds of times. That's called bootstrapping. And the most number of times when the sequence, when it is aligned in the same manner, you get more and more confidence that this is the right sequence. Okay. Because remember, there are millions of fragments which has to be stitched. Okay, cool. So this is what is done after fragmentation. So you can write down. And one very important thing before we do these fragments is when we isolate DNA from any human sample, we get some amount of DNA, but that's not enough. So what we will need is we will need to amplify each fragment of DNA, right? Each piece of DNA, we would like to make multiple copies so that when we sequence, we get multiple replicates of that sequence. because sometimes while sequencing, the machine can just have an error. Maybe it read A as T. If I just did it once, maybe I will end up having a wrong sequence. But if I did it 10 times and 9 out of 10 times it read T, and just one time it is reading as A, and one time it is reading as, just give me 1 second, people. Okay, am I audible, people? Sorry for the interruption. Am I audible?
Yes, yeah.
Okay, cool. I thought I lost you. Yes. What was I saying? Yes. So let's say I have one sample and I make a fragment, but if I just have one fragment of that part of DNA and I'm reading it, let's say this fragment 4 I am reading, and the machine read C as G or something by mistake. Okay, because mistakes can happen. It's a machine. It's not foolproof system. Even biology is not foolproof. But mistakes also happen when the cell is replicating the DNA. So this can happen. But we need multiple copies of every fragment, so that if there are 10 copies of a fragment, if I'm reading it 10 times, if 9 out of 10 times it is reading it as C, and one time it is reading it as G, I know that the actual base there is C and not G. That one time is a mistake. Do you understand? Yep.
Yes,
Is it clear?
yes,
Yeah.
yes.
So how will we amplify the DNA? So one way to amplify is to clone. So cloning. So cloning is done to amplify each piece of DNA fragment. Now, how to do that is using, how do we clone DNA? Any idea about cloning, DNA cloning? If I have this one piece of DNA, let's say this piece 4, GTCAATT, this one, and I want to make 10 copies of it. After I fragmented the DNA, I want to make copies of each fragment. Then what I can do is I can use a host that replicates very, very fast. And if I can insert my sequence that I want to clone inside a host genome, which replicates very, very fast and which can make millions of copies or hundreds of copies of my sequence of interest in its body, then I can use that as a host. So the commonly used hosts in gene cloning in biology are bacteria and yeast. Okay. And you know that yeast, what is yeast, people? Class 11th classification chapter. What is yeast? Animal, plant. Which kingdom it falls in? Quickly, come on. Yeast is a fungi. Okay. Kingdom fungi. And what is special about yeast? The special thing about yeast is that, yes, what is it, Aaron? Special thing about yeast?
It is, I don't remember.
It is? Sorry, I could not hear what you said after that.
I
It is
don't
what?
remember. I don't remember.
Okay, so it is the only unicellular eukaryote. Okay, it is a eukaryotic cell, but eukaryotes are mostly multicellular. Yeast is a unicellular eukaryote. So the advantage that you get to do research on when you do research on yeast or you use yeast is it is a single cell that you have to worry about, and it is a eukaryotic cell, right? Baker's yeast. So if you want to clone something which is compatible with the eukaryotic system better, you can use yeast as your host. And if you want something where it doesn't have to necessarily be a eukaryotic cell, even prokaryotic will work, then you can use bacteria, which is even more simpler to work with. So if you are using bacteria, then we make an artificial chromosome, which acts as a vector. Vector is basically anything that carries your gene of interest or your sequence. So the vector that we make using bacteria is called BAC, bacterial artificial chromosome. Write it down. For gene cloning, we can use two type of vectors. One is bacterial artificial chromosome, because we kind of synthesize it chemically. And the other one is YAC. It's called yeast artificial chromosome. Okay. And then once you have these multiple copies of every fragment, then you send it for sequencing. Okay, so every copy or every fragment gets sequenced multiple times, just for no errors are incorporated for surety. And then we use a machine which is called automated DNA sequencer. It cannot be done manually. So this machine works on the principle that was developed by Frederick Sanger. It's called Sanger sequencing. Frederick Sanger also developed the method to sequence amino acid sequence in a protein. But here we are sequencing the nitrogenous base sequence in a DNA. And how it is done, we will get into the details sometime later. That's beyond the scope of your syllabus at the moment, or we'll do it in biotechnology. I'll teach you, don't worry. But know for now that these samples go into a machine, and that machine tells you the sequence going each base. And it's very obvious to understand that, see, if you have 4 different bases, how you do the sequencing is nowadays is you tag a color with 4 different bases. So all the machine has to do is to see. If it sees red, it will read it as A. If it sees T, blue as T, green as G, and black as C. Okay, so in a DNA, you will have bases one after the other, right? If I keep it straight, you will have bases one after the other, right? So this will be red, then it will go to next layer, and it will keep reading. Okay. Make sense, everyone?
Yes.
Yeah. And I'm just explaining in very layman's language that they are color coded kind of. The principle is same, but the methodology is a little more complex. But just know that all these 4 bases are made chemically distinguishable so that the machine can distinguish. It's obvious. Otherwise, there is no other way, right? Yes. So then they are sequenced, and these fragments need to have overlapping regions present in them so that they can be aligned back together. Okay. Do you understand the methodology, everyone?
Yes, sir.
So this methodology, please make notes of it based on what you understood now from your textbook. Okay. And reach back to me if you have any issues regarding it. Any questions, Mariam and Iram? Iram, any question? Anything you want to ask?
No, sir.
Okay. And remember that there were 24 chromosomes that were sequenced from each sample, 22 autosomes, and X and the Y chromosome. Okay, perfect. So let's now talk about, once we know the sequence, now let's talk about DNA fingerprinting. DNA fingerprinting. Now, what do we mean by, there are 2 terms here. So forget about the first term because you know a lot about DNA now, right? What do we mean by fingerprinting? What is fingerprint? In our biometrics, we give the impression of our thumbs and fingers as one of our identifying features, right?
Yes. Pattern.
It's a pattern, it's a line pattern present on the surface, skin surface of our fingers, right? The tip of our fingers. Actually, it's present throughout our fingers, but evident, like obviously we can give impressions. You can also give your full hand impression, and that will also be different from other humans. So every human have a unique pattern on their fingertips, including the thumb tip. That's called the fingerprint. So the fingerprint is unique. No two people can have the same fingerprint. Okay. Now let me ask you a question. You know about twins, right?
Yes. Yes.
How many kinds of twins are there? How many types of twins are there?
So what do you mean by types?
Like all twins are like same kind of twins, or there are
No, they're
some
identical
twins?
twins.
Yes, right. So there are something we call as identical twins, and there are non-identical twins. What are non-identical twins? What are non-identical? These are, I'm brushing up some concepts of yours of genetics. Non-identical twins, reproduction and genetics both give rise to twins. How does a embryo form? Twins who don't look alike, Mariam says. Parker says, yes, Parker, you're correct. Twins who do not look alike are called non-identical twins. In the case of non-identical twins, you would like to note down, there are two different fertilizations that happen. Okay. Non-identical twins are formed because two sperms happen to fertilize two ovums at the same month inside a female. Both the ovums get fertilized by two different sperms and reach the uterus and gets implanted. They can get implanted at two different sites also in the uterus. Okay. One can be a boy, the other can be a girl also. The both can be boys, both can be girls, but they will not look exactly same to each other because they are non-identical. As the name says, they are formed from different ovums and different sperms. Identical twins is a rare phenomena where there is only one fertilization that happens. One sperm fertilizes one ovum, and as the ovum move towards the uterus from the fallopian tube, now you know all these things, right? Everything what I'm saying is familiar to you, all of you. Anyone?
Yes.
Anyone who's not following up, please stop me right there and ask me for some explanations. Okay. You have all the rights to raise your hand and ask for explanations in any class. Okay. Though I encourage not to miss classes, but if you have missed classes because of any issues, I'm not the kind of teacher who will punish you for that you were not present in the last class, so you cannot ask questions. Okay. You can, every class is a new slate. You can ask questions as many times as you want, even if it is the same question. So don't hesitate. In identical twins, one sperm fertilizes one ovum. When the zygote travels towards uterus to get embedded, when it becomes two cell from one cell. So from one cell stage, when it undergoes the first division, first cleavage division, and two cells are formed, because of some issues, two cells, instead of staying together, sticking to each other, they get separated. Now you have two small cells that got separated, but basically they had the same identical genome because they were coming from the same zygote. These two cells get separated and they can get implanted separately. They also start dividing separately. So two lump of cells are formed. And then these two will go on to form two entirely different bodies. But the genome in both the bodies will be same. So their sex will be same because it is already decided when the fertilization happened. Their complexion, hair color, hair texture, eye color, everything, they will be identical in every aspect of their morphology when they will be born, both morphology and anatomy at the time of birth. But my question is, do you think identical twins, what do you think? Do identical twins have the same fingerprint or do they have different fingerprints?
Different.
Different fingerprints? How is that possible then? Yes, Iram, how do you think it's possible? Their genetics is same, everything will be same. So how do you think fingerprints will be different? They have the same genes, they have the same chromosomes, they have the same alleles for every gene. Nothing is different. Actually, they were supposed to be one person, but just at the crucial junction of first cleavage division, the two cells got separated and developed into two different embryos. But basically, they are the same biologically. So will they have same fingerprint or different fingerprints?
I think same.
You think same fingerprints? Okay, Aaron. How many of you have you ever encountered around you among friends or families any identical twins?
Yes, sir.
Yeah.
Yes.
It's a rare phenomenon, but we are 8 billion humans. So even a rare phenomenon in 8 billion humans, you might have seen someone at least. I know of 2 identical twins in my life. Yeah, 3 actually. Yeah. They're not all my relatives, but they're very distant friends, mutuals of mutuals, but I know that they're there. So you must be knowing, right? Identical twins. Did you ever figured out if they have the same fingerprint? Can one use the other's passport and travel?
No, sir.
No, why?
No.
What is the then what if they have the same fingerprint? See, these are questions which comes from biology in our day-to-day life. We see it, we never ask. You never ask this question to yourself? Come on, I can't believe it. There are a lot of myths and misconceptions. People always ask questions to twins. I had an identical twin in one of the programs I was teaching in at my university for a month. And they were in school and their schoolmates used to ask them a lot of questions. Do you feel the tickling if I tickle this person? And so on, because there are a lot of myths around, also facts around identical twin, very, very common, prevalent in society. So you never asked if they have the same fingerprint or not? Okay, let me bust it for you. You should have. Please be curious around yourself. So even identical twins do not have the same fingerprints. They can have similar prints to a lot of, like it could be 60% similar, 70% similar, but it won't be identical because fingerprints are not a result of just genetics, just the genes, right? It's a developmental feature. You know what? If a person loses fingerprint, let's say the person loses the layer of the thumb or the skin in some accident or something, next time if it regenerates, the chances that it will have exactly the same fingerprint is also not 100%. There might be some variations when it regenerates it again. But those are very minor. We still consider them as identical. But identical twins also do not have the same fingerprint because fingerprint development depends on even the minor changes in the environment of the womb, pressure, force, movement of fluid around the baby when they were being formed can change your fingerprint. If you look at your finger and your hand very, very carefully, just do it at the moment right now. So hold it like open, straight open in front of you, your palm facing the face. And then if you try to clench your fist, you will see that most of the lines are where your hand and your fingers bend, right?
Yes,
Is it
sir.
correct or not? Yeah. So most of these features comes from how we close, open, and use our hands. And similarly, the fingerprints comes from very fine movement of fluid around the baby when the baby is developing inside the womb. So even though two twins are present in the same womb, the right side of the womb may not have exactly the same kind of factors and forces as the left side of the womb, and their fingerprints will be slightly different from each other. That's why even twins cannot use each other's passport and Aadhaar cards, driving license or voter's card, because they have biometrics, which is fingerprint. Okay. Even their eye color will be same, but fingerprints won't be. It's interesting, right?
Yes.
So from that fingerprint comes the word DNA fingerprinting. And DNA fingerprinting tries to achieve something similar. Can we use sequences of the DNA and identify differences in some specific regions? Okay. And can I say with 100% surety that this piece of DNA belongs to Iram, and this DNA belongs to me, and this DNA belongs to Parker, and this belongs to Mariam? Can I do that with 100% accuracy? That is the concept of DNA fingerprinting. Now you might be thinking that it doesn't look possible, but because I already told you that 99.9% of the sequence among all the humans, regardless of whether you live on the same continent or not, is same. So 99.9% of my sequence will be same as Iram sequence, even though we never have met our ancestors long back somewhere, millions of years ago might have been common, but not now. But still 99.9% sequence similarity will be there. It is that 0.1% that makes all the difference. And this is where we have to look. Okay. So there is something in the DNA in human genome, which is known as repetitive DNA. Okay. These are small stretches of DNA, which are repeated multiple times. Okay. And because they are repeated multiple times, they form a large portion of our DNA. Do you understand? Now,
Yes.
these pieces of repetitive DNA are called satellite DNA. Now, they can be on the basis of the length, they can be called macrosatellites and microsatellites and mini satellites. Okay. So depending on what type of base is present, if it is AT rich or it is GC rich, if the length is smaller or bigger, the number of repeating units are less or more, you can differentiate these satellite DNA regions into many categories. Microsatellites, which are very, very small repeating sequence, like let's say 20 to 10 to 20 bases repeating multiple times. Then mini satellites can have hundreds of bases repeating in the same pattern. So from 1 to 50, it will be a pattern. Then again, from 51 to 100, it will be the same pattern. Then 101 to 150, the same pattern. So, but basically, what is a satellite DNA? It is a repeating DNA that forms repetitive DNA sequence that forms the bulk of human genome. Okay. Now, now taking these satellite DNA into account, many a times a very small change. Okay. When I say very small, it can be a single base pair small. It's called SNPs. I gave you this as a homework in the last class. Single nucleotide polymorphism. Did you go back and read it? SNP, anyone could figure out what is SNP? As the name suggests, a single nucleotide changes randomly in this repeating sequence. Okay, somewhere. Now, it's like a needle in the haystack, but if we know where to look, so these regions, these satellite regions are very dense repeating units, which we know where they are present on which chromosome. We have mapped it. Every time you want to figure out that this piece of DNA belongs to which person, you just have to look at that small region of chromosome and not at the whole genome, because the whole genome, 99.9%, it will be the same. But if I look at just that part, and why does that part is different and still exist? Because that is a non-coding region. These repeating regions, repetitive DNA, are non-coding. Bulk of our DNA is non-coding. It does not code for RNA or proteins. So even if it mutates and accumulates, so suppose person X has two progenies, Y and Z, let's say, both both got the DNA, then they both had some mutations in their own genomes due to some reason, but that mutation was in this repetitive DNA region, the satellite region, and it is doing no harm because no gene is getting no protein coding gene is getting affected, so it has no consequence. It will just sit there because the mutation is in the non-coding region. Over time, as it will pass it to generations, the two brothers, their lineages will become different from each other in that 0.1% region in these repeating sequence. Is it clear, both of you? So suppose, is it clear or not? First thing.
Yes.
Now, so suppose I have two blood samples and I want to figure out, I did not label it now, I want to figure out which blood sample is mine and which blood sample is Parker's. What will I do? So I will take the DNA from, isolate the DNA from the blood samples, and I will ask I'll ask Parker, and I will also give another sample. We will take DNA from both the samples, and we will only look at this satellite region, let's say on chromosome number 14, this small region, where if I have any SNP, any single nucleotide polymorphism simply means multiple forms. So if I have any mutation in this region, it will for sure be different from Parker's. The repeating sequences will be different at just 1 or 2 points. So I will look at mine, and I will look at the pattern of mine, and I will look at the pattern of Parker's satellite DNA, and we will match it with the samples that we have, and we'll figure out, oh, this is mine and this is, it's like a code hidden within the overall similar kind of a coding. Do you understand?
Yes, sir.
Yeah. So I want you to come to figure 6.16. Let me pull it out for you as well. Open figure 6.16 from your textbooks, everyone. Let's see, where is that figure? Yes, yes. Could you find the figure?
Yes, sir.
Yeah. Okay, let me share the screen again. Yes. Can you all see the screen that I'm sharing?
Yes, sir.
Perfect. This is the figure, right?
Yes, sir.
Now let's do some Sherlock Holmes kind of a work. Okay, so look here. Now these kind of techniques are very helpful because they are used very widely in criminology and in forensics to solve cases. Like suppose there was a scene of crime where the victim's blood is there and so victim's DNA is there and another DNA is there, random DNA. Now that DNA, chances are it belongs to the criminal, right? So if you can figure out whose sample is that, then you know that who's the criminal. Or in cases of maternity and paternity claims that this person is the real biological father or not, we do the same kind of test. Now look here. I want to focus first on this part, okay, which I'm marking here as green. Don't go to this part. Look at this part. So this is a DNA from a crime scene, they say. Okay. So we got two samples, and sorry, yeah. So we got a DNA from a crime scene which has chromosome number 7, 2, and 16. Okay, so this is chromosome 7. It will be in a copy. This is chromosome 2. Again, it will be in a copy, and this is chromosome 16. And then we are looking for these short tandem repeats, the repetitive sequences, right? Which I told you, the satellite part of the satellite DNA. They are also called VNTR. VNTRs. It stands for variable number of tandem repeats. Okay, of a sequence. Now we exactly know where these repetitive sequences are in the genome. So we have to just look there. So let's say on chromosome 7, we find these number of short repeats. This is the representation: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 places. So chromosome 7, why there are 2 copies of chromosome 7? Because whoever this person is, he also or she also must have got this chromosome 7, one from the father and one from the mother, right? This is mother, one from the father. You understand? That's why there are 2.
Yes, sir.
So on one chromosome, there are 11 total tandem repeats, 0 to 11. On the other chromosome 7, there are 1, 2, 3, 4, 5, 6, 7, 8, 9 only. Similarly, on chromosome 7, there are some number of tandem repeats, which is 6, 8. And on the other homologous chromosome 7, there is just 7. So you can see what is color coded as the number of tandem repeats. Now, where we have DNA from 2 people are under suspicion, individual A and individual B. Okay. Now we will take similar samples from individual A. We took the blood sample of individual A and we took the blood sample of individual B, and only looked at that particular region of the chromosome. We don't have to look at the whole genome. So we looked at these tandem repeats of that. We looked for these tandem repeats in that particular region of chromosome 7. First, for individual A, this pattern came, and for individual B, this. And similarly, we got the patterns. This is just a representation in the gel format. So when you will run these, this is just a representation. But how do we know that these sequences are there? They have certain length. Okay. So when you run it on the gel electrophoresis, have you studied about gel electrophoresis? Anyone? Yes or no? Depending on that, I will explain.
No, sir.
No. Okay.
No.
Okay. So what happens in gel electrophoresis is we make a gel medium with small wells. Okay. So this gel is a thick gel. It has some thickness. It's like your bread. And this gel is made up of, agarose. So agarose are polymers. When they solidify, they form polymers cross-linked to each other. So they make a mesh-like network. Now, depending on what concentration you are using, you can either make the mesh very, very close to each other, or you can make the mesh far away from each other. So in this case, this is low concentration and this is high concentration. In high concentration, do you see that there are small holes between the mesh? Right?
Yes,
You
sir.
see everyone?
Yes.
But if the concentration is low, these holes will be bigger. Can you see this?
Yeah.
Yeah. I'm just representing is like this. It is more neat when you do the experiment. But you can see here the holes are small, here the holes are very, very big. So depending on what kind of gel we make, if there are bigger fragments, and if I load my DNA sample here, so I load in these wells, I don't know which belongs to which. And then because DNA is negatively charged, I will keep this gel in a buffer, in a water container, like it's a buffer container containing a liquid solution. And this liquid solution, I will attach a negative terminal and a positive terminal, and they will be connected to a battery. Now, when current will flow through this, because this is negative and this is positive and DNA is negatively charged, to which side it will be attracted to? This side, right? There will be a force on DNA molecule to go towards the positive terminal, right? When it will move towards positive terminal, it has to cross through these bushy mesh-like structure. Now, it is, just to give an analogy, I have a 2-year-old kid, I have a 10-year-old kid, then I have a 20-year-old person, and these 3 people are playing the same game where they have to cross through the bushes. So who do you think will easily cross through the bushes and who will find it difficult to cross through the bushes?
The 2-year-old kid.
2-year.
2-year will find it difficult or easy?
Easy.
Easy.
Why?
Because of its size.
He's small.
Yeah, because it's tiny, right? It can just crawl under the bush, and a 20-year-old will have to like curl up and do this, try to push the bush around and like that. So same thing happens for fragments of DNA. If the fragments are smaller, they will move faster and farther, and they will come here and form a band. If the fragments are bigger, they will stay closer to the well itself. Make sense? Yes or no?
Yes. Yes.
Yeah. Now let's analyze. You understood gel electrophoresis now. On the basis of charge, we make DNA move from negative terminal towards positive terminal, and through a mesh of agarose polymer. So now let's look here. Chromosome 7, on one chromosome, it had 11 repeating regions. Okay, so this is a very big region. So it will stay closer to the well. So this is the well where you loaded the DNA. So the closest one is the biggest one. Which is the smallest one? On chromosome 16, there are just 2 repeating units, 2 tandem repeats. That is the smallest. So this will go the farthest from the well. Make sense? They have arranged it in the order of
Yes.
decreasing size. So you also see the pattern like that. Make sense, everyone?
Yes, sir.
Now, when you run individual B's DNA, the 2 suspects' DNA, you will again get a pattern. Here you are getting individual B's pattern exactly same as the DNA which was found from the crime scene. So what does it tell you? Of course, it tells you that B is the criminal, but what does it tell you biologically? That the DNA belongs to person B because only B has the same tandem repeats with the same sequence at the exact same location in its genome. Right? No other person can have that because these are non-coding sequences. They accumulate change and they do not cause any problem. So they are carried forward in the genome. And this is what is called DNA fingerprinting. So we actually can, as accurately as fingerprints, we can do DNA fingerprinting. This technique is called DNA test in layman's term. You must have heard this test that you can do a DNA test to figure out whose father is whose father. Correct?
Yes.
Now, this is for the person who is a suspect. How can we do it for maternity or paternity test? Similar. So if I'm doing it for paternity test, only one chromosome will show the same sequence, not the other, because suppose person A wants to figure out who is his father, and there are 2 or 3 suspects to figure out from, or in cases where there is a natural calamity and you are not able to identify the diseased person's body, then you go for a DNA testing to figure out whose body is this. Then in that case, you take the parent's blood in that diseased person's tissue sample, and then you look for similarities, right? So half of its genome will match father's genome in terms of repeat sequences because that DNA came from father, and half will match the mother's genome because that came from mother. Do you understand?
Yes, sir.
Yeah. Now that person himself can also accumulate a few changes, but that will be less. It's not possible that you take the DNA from your parents and you change it yourself so much that it looks nothing like your parents. That's not possible. You accumulate one or two things in it, but it will still have a lot of tandem repeats. Most of the tandem repeats will be same as your parents. So this is how DNA fingerprinting works. And it has a very wide application in population genetics, understanding diversity in species, and also to solve criminology and forensic cases, and for paternity and maternity claims. Is this part clear to all of you?
Yes.
Anyone has any doubts? With this, we come to the end of this chapter. So I want you to, there's one paragraph on this, which talks about repetitive DNA. So what is repetitive DNA? It is specific regions in DNA sequences which have repetitive sequences. These are small stretch of DNA which are repeated multiple times. They are called repetitive DNA. What is a satellite DNA? These repetitive DNA, which they are separated from the bulk DNA as different peak. So when you do the DNA centrifugation, like Iram was suggesting, one way to do it is you can centrifuge. So in gel electrophoresis, you are pushing them to move through something. In centrifuge, you are just rotating them fast enough that the bigger ones, the bigger sized fragments will settle down faster. So in centrifugation gradient, you will see that the bulk of the DNA is it just settles down, but some small peaks which are of the repetitive DNA are called satellite DNA. Then third is polymorphism. What is polymorphism? It is difference in a particular base or a sequence stretch, a small sequence stretch that arises due to mutation. So polymorphism happens because of mutations in the genome, and these mutations, why are they being selected and carried forward? Because they are not happening in any crucial gene. If these mutations happen in a crucial gene, they will cause disease and the person will die, or the person will not form as an embryo at all. But since they are happening in repetitive sequences in the satellite regions, they are accumulated and carried forward, but they are helpful for us to do DNA fingerprinting. Okay, so they are inheritable. And then what is VNTR, variable number of tandem repeats? So it is, we actually can count how many tandem repeats are present in a particular stretch of a DNA. So the technique of DNA fingerprinting, which uses satellite DNA as probe, is called the VNTR. So this probe is called VNTR, variable number of tandem repeats. And this technique was actually, I think it was developed by Alec Jeffrey, yes. So Alec Jeffrey developed DNA fingerprinting technique where you can use a probe. Probe is nothing but an artificially created sequence, which you know will go and bind to your actual DNA, and you can then amplify it. So just read this part from the textbook and let me know if you have any problems or doubts or queries. If you understand this figure here, you understood the gist of it. Rest is making notes, and I'm saving some time by not dictating it because it's straightforward in NCERT. But if you want me to in next class, I can do it. Not a problem. But first, you do it yourself. Okay, everyone? Anyone has any doubts to ask? Iram? Yes, you want to say something?
No, sir. I think okay.
Okay, perfect. So next, what we are going to start is plant reproduction. So sexual reproduction in flowering plants. We will understand plants and then we'll go to evolution. So what I want you to do tomorrow, today, not today, then tomorrow, before you come back day after tomorrow, is you have to understand what are the sexual organs of a plant. Okay, and we will focus only on flowering plants. So from class 11th, you know that all plants are not flowering plants, right?
Yes, sir.
So what is the name given to flowering plants?
Angiosperms.
Angiosperms. And angiosperms are the most abundant on the planet, right? Even grass, it starts with grass. Grass is also an angiosperm. So angiosperms make flower, and flowers are nothing but sexual organs of plants. So I want you to go and read the structure of a flower. What is the male part? What is the female part? And then I will start with how fertilization happens in plants, and then we'll go further. Is it clear?
Yes, sir.
Yeah. So this chapter might feel a little boring if you are not interested in plants or botany. But see, it is not just boring for you. It's boring for majority of biology students who want to become doctors or are interested in animal system and human system and not in plant system. But believe me, ranks and merits are decided on the basis of these chapters mostly, because mostly we all are good with animals, humans, their diseases, genetics, evolution. But then botany is one part which is boring, so we don't pay attention to. Okay, I'm not interested in plants. Anyways, when I will become a doctor, I will not be treating plants. You are right. But to become that, you have to qualify exams, right? That's the hard hitting truth. But so I'm just preparing you that if you pay attention to plants and take it just out of curiosity, try to understand, oh, these are amazing systems. Don't you think plants are more amazing than animals? Because see, one, they don't have a nervous system, they don't have a brain. Two, they don't have a blood circulatory system like us, but they have a different kind of a system. So no brains, they cannot move. Most of them cannot move. Some plants can, but those are exceptions. But still, they are present everywhere. They are very successful on the planet. When you go to a forest, you have to work hard to find a tiger, but you don't have to work hard to find a tree. Trees are everywhere. Animals are not. Right?
Yes, sir.
So, and no one, now we have started this drive. So we have to protect plants, save trees initiative and all. But when humans were not there, this whole planet was like a jungle. Lots of trees and plants growing everywhere with no one's help. No one was seeding them. Like when I say no one, I talk about humans mostly. But still plants are very successful in evolution. So why? Because they have evolved some